Hello everyone, I'm Koyan and today we are going to learn how to answer question related to Form 4 Chapter 5, which is Metabolism and Enzyme. So let's take a look at this question right here. Diagram 2.1 shows the action of enzyme X on substrate maltose. So yeah, maltose is basically the substrate, but if they didn't tell you, you can also basically know that this is the substrate. And the enzyme one, which is slightly larger in size, look like a Pac-Man. So for this one, you have to identify what is this enzyme X. State the name of enzyme X. It's an enzyme, but what is the specific name of it? So since they already given you the specific name for this example, which is maltose, so maltose is actually a disaccharide, right? So disaccharide is actually can be broken down to monosaccharide, broken down to these two monosaccharide. So if you can recall the equation, when maltose is being broken down, or you can say as hydrolyzed to be precise, it will form glucose and glucose plus glucose. So P is basically two molecules of glucose. So what is the enzyme X? Enzyme X is actually the enzyme which ends with ASE. So it is maltase. Most of the enzyme name are ended with ASE. Yeah. So the enzyme is maltase and name of molecule P is basically glucose. Now let's take a look at the question 2 based on diagram 2.1 state 2 characteristic of enzyme X. So a lot of students, they didn't really focus on this part based on diagram 2.1. A lot of students really know inside from their brain, they really know all the characteristic of enzyme, then they just write it out. Okay, characteristic of enzyme, there are actually a lot of them, but you have to actually base on this diagram. So in normal situation, if they just ask you the characteristic of enzyme, you can actually just write enzyme is the one that speed up a biochemical reaction. The enzyme action is specific. It required in small quantity. The structure of an enzyme is unchanged after the reaction. The enzyme requires a cofactor to work at maximum rate or yeah. And then yeah, inhibitor is the one that inhibit the action of enzyme, or it can act rapidly, or enzyme is reusable. The enzyme action is reversible. However, this one they need you to base on diagram 2.1. So you have to be specific for this type of situation. You have to see what is the characteristic you can add on to it that actually talk about this diagram. So if you take a look at this part, you can see that this huge arrow right here, okay, since it hydrolyzed already into two molecules of glucose, it can be a reversible action. It can reverse back. It can still form back to this maltose through condensation process. So this is actually called that it is a reversible action. So this is one of the characteristic of the enzyme. So reaction catalyzed by enzyme are reversible. Other than that, you can see that these maltose have this specific shape, and then the active site also have the same shape so that they can be joined together and be broken down. So this actually means that it is a highly specific action. So the next characteristic you can actually write is highly specific in action. And okay, by looking at this part, you realize, oh, you have written down reversible and then it is a highly specific. What about rapidly? Rapidly, the diagram didn't really show that it's rapid, right? No, you can't write it out. Okay, reusable and reversible is almost the uh, same concept, but it's better to write reversible because this is based on this arrow, which is clearly stated right here. And then speed up, you can't really see that it's speed up, right? And small quantities, yeah, you didn't see that they use it in the small quantities. Cofactor inhibitor, they didn't even involve it in this diagram. But another thing that they show you is the structure. Does it change? The enzyme structure doesn't change, right? So it means that, yeah, it remains unchanged. So enzyme remains unchanged. Enzyme structure, or you can write are not destroyed lah. Are not destroyed after reaction. Or you want to write it as remain unchanged. Also acceptable. So yeah, these are the 3 points you can actually write because it is based on the diagram 2.1. A lot of students fall the trap on this part. Okay, so let's move on to this question right here, pink salmon. So they basically using an enzyme immobilization technology that used in fish canning industry to produce a skinless fish. So how the technology helps in industry? So they are actually wanting you to emphasize the immobilization technology. And they don't want you to really like talk about how you use the protease enzyme to produce, yeah, you can produce a skinless fish, but what about when they apply this technology, how it actually enhance its function? So you can actually state out what is the enzyme used lah to produce skinless fish. So you are basically using a proteins enzyme. However, how are you going to explain the immobilization technology? So immobilization technology, right, is actually helping the enzyme to overcome this situation where it is very sensitive to temperature and the pH change. So originally, enzyme is very sensitive to the changes in pH and temperature. So when the pH or temperature deviate a bit, yeah, it won't be able to function in an optimal condition. So they use this enzyme immobilization technology to increase their resistance, increase their resistance to the changes in the temperature especially lah, so that it can function at the most optimum state most of the time. Because if you take a look at the graph, um, let's say for this part, okay, initially it functioned at the optimum state at that, yeah, basically this range, but it's just a very short range. Let's say only like 20 degrees Celsius right here, and only that specific range where it functioned the most optimum. So if you use this enzyme immobilization technology, it makes it having the resistance to, I mean, prevents it from denaturing easily at the high temperature. So what do they add inside this enzyme to make it to increase the resistance to the temperature is actually adding something insoluble, or you can say inert lah. So protease enzyme combines with in inert, or you can write it as insoluble material, insoluble substance. So this will cause enzyme to to have resistance in, sorry, to cause, sorry, to cause the enzyme to increase its resistance to temperature. Because normally when the production of this, they normally produce, the surrounding temperature normally is very hot. But- So yeah, basically they want it to increase its resistance to the changes in temperature. So that other than that, the enzyme molecule will remain in the same position throughout the whole reaction. Okay, in normal situation, right, when the enzyme catalyze the substrate, okay, the product it produce, right, normally it will eventually it tends to stick with it still tends to stick inside the enzyme there, like very hard to leave lah. But if you use this enzyme immobilized technology, the product and the enzyme are easily separated lah. So that's another point you can actually mention for this enzyme immobilization technology. And another point you can also mention is, what does the protease enzyme do? It actually can remove the fish skin. So yeah, that's all. So most importantly, this enzyme immobilization technology is where it combines the enzyme with the inert substance, and then causing this enzyme to be more resistant to the change in the temperature. And then the enzyme will remain in the same position, and the end product and the enzyme will be easily separated lah after the reaction. And another thing you want to add on, since this one is more specific towards this protease enzyme, you can also add on this point. Okay, now let's continue to the next question. So it's from the MRSM paper. So it actually shows the stages in enzyme J reaction based on the log key hypothesis. So based on diagram 1, name J and M. So J is basically an enzyme, but you have to be specific what type of enzyme is that. And M is the product, but what is the product in specific? Because biology, they need it to be specific since they already given you something, a substrate that is a lactose. So when the lactose are being hydrolyzed, it will form glucose and galactose. Yeah, sorry, cannot see. So what is the enzyme that catalyzes this reaction? Is the enzyme that ends with ASE, so it's lactase. So J is a lactase enzyme and M is the glucose. You can either write glucose or galactose, they will accept both of them lah. You just write one of them, not two lah. Because they only point out one only, so you just write out one of them. So what is the structure L? So structure L is actually an enzyme substrate complex. But can you actually write enzyme substrate complex? For this situation, no, because it's very specific that this one is a lactose, this one is a lactase. So when you want to write enzyme substrate complex, in specific, it is a lactase lactose complex. So what is the type of metabolism involved in the formation of structure M, which is this product? the monosaccharide. So what is this metabolism name? Can you call it as hydrolysis? No, it's not hydrolysis. Because type of metabolism, they divided into two types. One is anabolism, another one is catabolism. Anabolism is basically you build up things, you synthesize something, you produce something lah. You build out something complex from something simpler. But catabolism, you see, you cut it out. So catabolism means you break it down. So you are breaking down. So this one is actually breaking down or building up. They are actually breaking down from a complex, which is disaccharide, to become a monosaccharide. So this is a catabolism. Then the next one is actually explain why J does not act on substrate K at stage 2. So can you see the J? Where's the J? Oh yeah, the enzyme, why it cannot act on this K? Why it only acts on this lactose? So basically it's because their shape is not fit together lah. And yeah, you have to mention what is the characteristic of that enzyme. It is very specific, right? Which part is specific? The active site that is specific. So you can write J has specific active site. You must mention the active site is very specific. That's the reason why the K cannot complement to it, cannot fit to the active site. So substrate K, that's the reason why substrate K does not fit or complement, if you want a better word right here, complement to active site of the enzyme of the J. So the means it cannot bind to the active site. Substrate K cannot bind to uh, I think you have to add on this really better. The shape of substrate K is not complement or is not complement. Sorry, I rephrased my sentence. It's not complement to the active site of J. So that's the reason why substrate K it cannot bind. So this word is important, cannot bind to active site of J. Because they have to be complement with each other. Then they can only be binded together to form enzyme substrate complex and product will be formed. But this substrate K, basically the shape of it, it's not suitable with the active site of J. Moving on to this Kelantan paper, so it's basically a production of the extracellular enzyme because it is being transported out of the cell to be used out of the cell lah. So there are actually two types of enzyme. So the first one I mentioned already, it is the extracellular enzyme. It will be transported out of the cell to be used outside of the cell. And there are also enzymes like the hexokinase enzyme. Yeah, it's actually mentioned in textbook, but it's very small statement right there. So one of the example is that. So it's an intracellular enzyme. So it actually will stay in the cell to be used lah. But this one will be transported out of the cell. This one will stay inside the cell. So intra and extracellular enzyme. So for this one, they want you to label the component involved in proteins transport as X in diagram 2.1. So what is the component that involved in the transportation of protein? First of all, the protein will be synthesized by the ribosome. So all this black dot present on the rough endoplasmic reticulum surface is the ribosome. So who is the one transport the protein synthesized by ribosome? It is obviously the rough ER. So they will transport through this transport vesicle to the Golgi apparatus, and then the Golgi apparatus will modify it to become enzyme. Then, yeah, it will bud off, and then it will become a secretory vesicle to be transported out from the cell. And yeah, so it is the rough endoplasmic reticulum. So when you want to label, you point it out and then move your hand. Okay, use your ruler and label it as X. You don't have to mention what is it specifically. So what is the type of enzyme? It is an extracellular enzyme. You state, then you only explain. So it is synthesized because it will be transported out of the cell. That's the main key word why it is called an extracellular enzyme. So it is synthesized size in cell and transported out of the cell to be used. So the main thing is it is transported out of the cell to be used. And it is an extracellular enzyme. That's how you get the 2 marks right here. And moving on for this lactose-free milk. Okay, people who have babies who have lactose intolerance means their body probably lack of this lactase enzyme or can't produce much. Yeah, so lactose intolerance can be caused by baby. So since they are not able, they have very less or maybe don't even have this lactase enzyme. So when they don't have it, it's very hard for them to break it down now. That break the lactose down. That's why it's called lactose intolerance, and it will lead to diarrhea in most of the situations, stomach pain. So overcome it, they use an enzyme to produce this lactose-free milk. So basically, right, the reason why this child will get diarrhea is because in a normal situation, we drink the milk. The milk sugar is actually lactose, not galactose, because it will only later on it will only be uh hydrolyzed into plus water. It will only be hydrolyzed to the glucose and galactose. So supposingly when we drink the sugar is still in a lactose form, which is a disaccharide form. But our body is the one that in charge to help to break this lactose down to be glucose and galactose. Okay. However, for the people that lack of this lactase enzyme that have lactose intolerance, you can imagine what happened to their large intestine right here, why it will lead to, yeah, it's a very terrible drawing, why it will lead to diarrhea is probably because, let me zoom in the huge large intestine right here. Okay, yeah, it's basically a lot of, okay, so if you zoom in the large intestine, the drawing is terrible. Okay. When you zoom in the large intestine, since they don't have this, they are not able to form glucose and galactose to be used by the body, to be absorbed by the body. So lactose, they are not, I mean, they can't be broken down to simpler substance to be absorbed. So the lactose still stays in their large intestine. So when there's a accumulation of this lactose, it will cause means that it has higher solute concentration than here has a lower water potential. So the water from the epithelial cell right there, yeah, it will actually diffuse out to the large intestine. So when here we eventually, because here got high water potential, go to the lower water potential. So eventually the feces will become extremely watery. That's the reason why it will have diarrhea. Because the lactose is unable to be broken down to be absorbed by the body. So yeah, after explaining so long, basically, how are you going to state the function of enzyme? So basically, this enzyme, it will hydrolyze lactose into glucose and galactose. So yeah, this is basically the function of enzyme. So you might be wondering, why some situation I have to write with the addition of water? Do I actually need to write it? Okay, when you write hydrolyze means you already stating that you are breaking down lactose with water, with the water. Because you already mentioned right here hydrolyze. Not only break down, you also write this hydro in front, yeah, means breaking down with water lah. But if you say break down, so lactase enzyme break down lactose into glucose and galactose, it may be incomplete because you have to write with addition of water. So it's better to use this hydrolyzed word. So you just clearly stated that it break down using water. Okay, so let's move on. I mean, basically now the lactase enzyme, it break down this supposing this milk got this lactose, so lactase break down it. So now the milk doesn't consist of lactose already. It consists of glucose and galactose to be consumed by lactose intolerant child. Okay. Okay. Biodetergent. Application of enzyme in daily life. So use of enzyme causes laundry to be cleaner, does not cause irritation to skin, and can maintain sustainability of ecosystem. The enzyme was used to produce through this immobilized enzyme technology. So how does this immobilized enzyme technology help the enzyme to improve its function? So it's just almost the same explanation like just now. So basically, enzyme will combine with the inert and insoluble substances, and this view enzyme has increased its resistance, has higher resistance to the pH and temperature. To be specific, to the changes in pH and temperature. You want to write temperature also can, pH also can lah. And then the enzyme will remain its original position throughout the catalytic process, and the enzyme are easily separated from the product. So these are the points, most important points for you to mention when it comes to enzyme immobilization technology that we learn in this SPM syllabus. You combine it with inert and insoluble substance so that it have higher resistance to change in pH and temperature. Then this enzyme will remain in the same position throughout the whole process, whole catalytic process. And other than that, other than remaining in its position, the product and the enzyme are easily separated. Ta-da! Okay, these are the 4 points for you. So next question is showing a metabolism process that occurs in a plant. So this is basically photosynthesis because they use sunlight and then they absorb carbon dioxide and produce this oxygen as a metabolic byproduct. So what is the type of metabolism? Type of metabolism got 2 types. One is anabolism, one is catabolism. So anabolism is the one that build up or synthesize something, synthesize a complex substance. And then catabolism is the one that break down the complex substance to be simple substance. So do you think this one is actually building up or it is breaking down? So obviously it actually what's the main thing of photosynthesis for a plant? They actually require photosynthesis to produce its own food, which is the glucose. So carbon dioxide plus water with the help of sunlight and the chlorophyll to produce glucose and oxygen. So they are actually trying to make glucose. So it is building up blood. So it is anabolism. And what is the product? The product is the glucose. Because glucose is the main product the plant actually required to use. Oxygen, it is also a product, just that it will be released to the surrounding because oxygen is actually re, uh not required for this plant lah, so they just release to the surrounding. So it's actually called a metabolic byproduct. So how do you explain this anabolism? So anabolism is basically a process where you synthesize complex substance. Synthesize would be a better word compared to build up, right? So anabolism is a process of synthesizing complex subs substance or molecules from simple molecule. So it's better to write simple because they sometimes may reject it if you write synthesize large molecules from small molecule, they will actually reject your answer lah. So please don't write, okay, you cannot write large, you have to say it as complex lah. And for simple, you cannot write it as small, they will reject your answer. So rather than synthesizing it, it actually will absorb energy. Catabolism process, it will release energy. But anabolism process, it will absorb energy. So that's another characteristic of this anabolism process. Okay, so moving on. The necessity of enzyme in metabolism is to speed up the biochemical reaction in a cell. So 3.2 shows effect of enzyme on the activation energy of the reaction. So I know a lot of students hated to see biology graph because it's hard to understand. No, it's not really that hard lah, basically. Okay, imagine every type of reaction, right? They have this thing called the activation energy. So the reactants actually have to overcome this activation energy in order to produce a product, in order for the reaction to occur. So imagine you are using, all the, let's say now, you want to do, let me think. Okay, let's say you eat something and then but you don't have an enzyme lah. So let's say you eat a piece of bread and then the starch will be broken down into the maltose. Okay, so supposingly we know that it will be amylase is the one that help out to break down the starch into the maltose to a simpler substance. But what about when there is no presence of enzyme? Will the reaction actually occur? Yes, it will actually occur. However, it will occur at very slow speed. So without the help of enzyme, the reaction will still occur, just that at a very slow speed. That's the reason why we state that enzyme is a biological catalyst that speed up biochemical reaction. So without enzyme, yes, biochemical reactions still can occur, just that it will occur at extremely slow rate. the starch we put down need like maybe days to be broken down. But if you use enzyme maybe in hours, then yeah, you get to break down already. Because enzyme is biological catalyst that speed up biochemical reaction. So how do they act on to it? Is basically by lowering down the activation energy. So without enzyme, it's quite hard to achieve the activation energy, but it still can achieve need a very longer time. So imagine you are rolling a ball lah from this reactant. You have to roll it so hard up the hill only can go down to produce a product. But this enzyme, it actually have this usage to lower down the activation energy. So let's say you are rolling the ball. It's so easy because it is a shorter height lah. Okay, so yeah, basically it will be more easy and more faster. So this is how enzyme works lah. So what is actually the meaning of activation energy? It is actually the energy required for the reaction to occur. So energy required to break the bond in substrate molecule before reaction can occur. Okay, so how the enzyme affects the rate of reaction, but you have to base on this diagram lah, how it can affect the rate of reaction. So first of all, how it can actually speed up the biochemical process. It actually lower down the activation energy, right? Initially it's this high, becomes so low. So it will enzyme reduce activation energy of the reaction so that the reaction can be speed up. That's the reason why the production rate will increase lah, or you can say increase the number of products from now. Because more substrate able to overcome this low activation energy to form products. So, or you can write it as more products for. Okay, moving on to the next question right here. Yeah, there's actually no more next question. Okay, so yeah, that's all for today. And I will see you in the next chapter. So thank you so much for watching and I'll see you. Bye bye.